Hello everyone, myself Jehrin J, Department of Biology. Today we are going to discuss about a new topic, Nucleolus and Ribosome from cell biology. Nucleus. Nucleus is a membrane-bound structure that connects cells' hierarchy information and controls the cell growth and the reproduction. It is the CPU of this cell. Nucleus is separated from cytoplasm by nuclear membrane. It regulates the integrity of gene expression, and so we can say that it is a control center of the cell. It is a specialized double bound membrane protoplasmic body that contains genetic information and also control the cellular metabolism and transmission to the posterity. Robert Brown is the scientist who first studied the nucleus. As we say, nucleus is consists of a nuclear envelope, nuclear pores, and nucleolus in the center. Nucleus is a uninucleated and possesses single nucleus. Paramecium has two nuclei, macronucleus of controlling metabolic activities, organisms, and micronucleus possess hereditary informations. Some cells such as multinucleated or polynucleated condition is found in some of the cells of bone marrow, striated muscles, several fungi, algae, latex vessels. Multinucleated animals are called syncytial cells, while in plants and fungi they are cyanotic cells. A cellular slime mold have multinucleated protoplasmic body called plasmodium. Approximately, the nucleus is present at the central region of the cytoplasm. As we already discussed about the structure of nucleus, it contains a nuclear envelope, nucleoplasm, nuclear matrix, chromatin, and nucleus. Now let's see each of the components in detail. Nuclear envelope. It is a outermost bound structure. Nuclear envelope separates the nucleus from cytoplasm, made up of lipoprotein and trilaminar membrane. It consists of 60 to 98 thickness. Is smooth in structure. Outer membrane may be smooth or its cytoplasmic surface may bear ribosome-like rough endoplasmic reticulum. The two-membrane nuclear envelope is separated through electron-transparent perinuclear space. The outer membrane is always connected or often connected to endoplasmic reticulum. Nuclear envelope contains large number of pores or perforations. It is a complex structure, may have diaphragm, septum, plug of nuclear dense material, nucleoplasmic annuli. Annuli are the circular structure around the pore. The pores and the annuli form a more pore complex called annulated pore. It consists of 9 cylinders, 1 central, 8 peripheral. Consists of network of granules and filaments. Nuclear pore control the passage of substance inside and outside the nucleus, example from RNAs or from ribosome or to protein during translation, transcriptions, etc. Here are the structure of nuclear membrane, perinuclear space and nuclear inner membrane, and we can also see the pore bulb, annulated pore and septum. Next, let's see what is nucleoplasm. It's a transparent semi-fluid colloidal substance filled in the nucleus, contain nucleosides and enzymes. It consists of certain enzymes, that is DNA polymerase, RNA polymerase, nucleoside phosphorylase. These are the enzyme machineries required for the synthesis of DNA, RNA, such as in replication, transcriptions, etc. Nuclear matrix is a network of fine fiber as a protein function as a scaffolding for chromatin. Dense fibrous layer is called nuclear lamina. Terminal end of chromatin fiber or telomere is embedded in nuclear or nuclear lamina. Nuclear consists of two types of intermediate filaments, that is lamin A and lamin B. Nuclear matrix and nuclear lamellae form scaffolding for chromatin attachment site of telomeric part, mechanical strength to nuclear envelope, and components of nuclear pore complex. These are the functions of the nuclear matrix and nuclear lamina. Next is chromatin. It is a genetic material. The DNA protein fibrillar complex is named because of the ability to get stained. They can get stained and they also form fine overlapping coil fibers which appear to produce a large network called chromatin reticulum. Chromatin fibers are distributed throughout the nucleoplasm. They are differentiated into two regions, euchromatin and heterochromatin. Euchromatin is narrow, light stained region, whereas heterochromatin is a dark stained, condensed granular region. Whole chromatin is not functional. Generally, only a portion of chromatin which is associated with acid protein take part in transcription or the formation of RNA during the prophase of nuclear division. The chromatin fibers condensed to form definite number of thread-like structure is called chromosome. Chromosome is the genetic material. Nucleolus, first discovered by Fonte in 1781 and has described by Wagner and provided the present name by Bowman in 1840. Is a naked, round, slightly irregular structure is attached to chromatin at a specific region called nuclear organizer region or NOR. Commonly 1 to 4 nuclei are found in nucleus. Up to 1600 nuclei are reported in oocytes of xenopus. Nucleolus has 4 components: amorphous matrix, granular part, fibrillar protein and chromatin. Amorphous matrix is a homogeneous brown substance of the nucleus. Matrix is formed of protein. Granular protein, it consists of granules of size 150 to 200, which lie scattered in the amorphous matrix. The granular upper form of protein and RNA of ratio 2:1. They are believed to be precursor of ribosome. Fibrillar protein portion. It is formed of a large number of small fibrils that are 50 so long. The fibrils are made up of both protein and RNA are believed to be precursor of granules. Chromatin portion. It is a part of chromatin which is associated with nucleolus. Depending upon its position, nucleolar chromatins of two types, perinucleolar and intranucleolar. Perinucleolar chromatin lies around the periphery of nucleus. It gives rise to ingrowths or tuberculi which produce intranuclear chromatin. Nucleolus is a principal site for the development of ribosomal RNAs, center for the formation of ribosome components. Nucleolus stores nucleoprotein, the same as synthesis in cytoplasm, essential for spindle fiber formation during nuclear division. Now let's see the functions of nucleus. Nucleus is essential and the main center of the cell is an integral part of eukaryotic cell, stores genetic information in its DNA molecule and passes it to the generation and controls cellular activities. Chromatin nucleus contains certain hereditary material called chromatin. Chromatin is a DNA protein complex. It is made up of number of fine fibers that condense to form chromosome. Number of chromosome is fixed for a species. They bear genes. It consists of functions, genetic information, cellular activities, ribosome, variation, cell growth and maintenance, cell differentiation, cell replication. Now let's see the another cellular component, that is ribosome. Ribosome are naked ribonucleoprotein or protoplasmic particle of length 200 to 340 A and diameter of 170 to 240 A. Ribosomes are popularly known as protein factories. They are subspherical in outline. Each ribosome consists of two unequal subunits, that is larger domain and the smaller domain. The large subunit has protuberance, a ridge and stalk, and the small subunit possesses a platform cleft head and base. Is about half the size of the large subunit. The smaller subunit fit over the larger one at one end like a cap. Using Mg2+, dissociate give about the strength of ribosome to come together and to form a dimer. Ribosome may occur singly as monosome in rosettes and helical group called polyribosomes in formation of mRNA. The different ribosomes of polyribosome are connected by messenger RNA. The maintenance of polyribosome requires energy. Size of the ribosome is determined by sedimentation. Organelle ribosome of 70S, but in mammalian mitochondria they have sedimentation coefficient 55S. Ribosomes have 4 specific attachment: mRNA binding site, amino acyl site for the binding of newly arrived amino acid carrying tRNA, P site, that is peptidyl site with tRNA carrying growing polypeptide, E site or excite site for the free tRNA before it leaves the ribosome. 80S ribosomes are synthesized inside the nucleus. Their subunits come off the nucleus and become operational in cytoplasm. Chemically, a ribosome is made up of 2-part protein and rRNA. 30 subunit of 70S ribosome possess 21 protein molecule and 16S ribosomal RNA. 60S subunit and 80S subunit has 14 protein molecule and 3 type of rRNA, that is 28S, 5.8S, 5S, 50S subunit of 70S ribosome contain 34 protein molecule and 2 type of RNA, 23S and 5S. Protein are both structural and enzymatic. The main functions of ribosome are help in protein factories, free attached ribosome, enzymes and factories, rRNA, mRNA and protection. That is, it helps in translation. Mainly, it have a great role in translation.